At this point in the course, we have completed our discussion of proteins regarding their structure and their function. We are now going to pivot towards the remaining biomolecules that we need to talk about in this course, which include carbohydrates, lipids, nucleic acids. We are first going to begin with carbohydrates, and as you'll see going forward, this was true for proteins as well, but as you'll see going forward, we are going to continue the general theme of talking about the building block first, and then start connecting the building blocks together into the larger polymer. In this conversation, the building block is going to be the monosaccharide, and the polymer is going to be the polysaccharide. So we will first talk about the monosaccharide properties, and then start tethering the monosaccharides together into the larger polysaccharide polymer. Before we talk about the details of carbohydrates, what they look like, what they do, here's the big picture. Here's the why you should care. Carbohydrates, simply put, are the most abundant biomolecule on Earth. You'll find that photosynthesis, just the act of photosynthesis, annually converts more than 100 billion metric tons of CO2 and water into cellulose and other plant products. So if you look around, you see plants. Carbohydrates are going to be a major component there. Outside of that, you and I, we oxidize carbohydrates in glycolysis, which is a very central energy yielding pathway in non-photosynthetic organisms. So glycolysis, carbohydrate oxidation, is critical to the production of ATP under anaerobic conditions. So you are constantly using this in muscle tissues as oxygen is being depleted, as you transition from aerobic to anaerobic metabolism. Carbohydrate polymers also serve structural and functional roles. Until the 1960s, carbohydrates were thought to really only have passive roles in the cell as energy sources and as structural materials here and there. When I say structural, think about cell membranes, cell walls. You'll see carbohydrates show up there. Carbohydrates, though they do not catalyze chemical reactions like enzymes do. They also don't replicate themselves like nucleic acids. So, really, they're very different from the other biomolecules that we will talk about. Proteins, enzymes are catalytically relevant, and carbohydrates don't really do those sorts of things. They're not built according to some sort of genetic blueprint like nucleic acids and proteins, and for that reason, they tend to be much more heterogeneous in both size and building block composition relative to the other biomolecules. However, it is this heterogeneity that is the fundamental driver of carbohydrate biological activity. The apparently haphazard arrangement of carbohydrates on the surface of cells are key to recognition events between proteins. Think protein cell recognition or cell recognition. And this is also important in the immune response. You'll see that carbohydrates are very important when it comes to immune responses. A very big example of this right now is the carbohydrates that show up on the coronavirus, the SARS-CoV-2 spike protein. You'll see that carbohydrates are important to its function. Strip off the carbohydrates and the function is not quite the same. So though carbohydrates themselves are not catalytic, they do facilitate catalysis and physiologic events for other biological molecules. Chemically speaking, carbohydrates are fairly straightforward molecules. They are simply polyhydroxy molecules that contain a single aldehyde or ketone functional group. So this will be important later on because the aldehyde or ketone, that functionality is how we define, how we classify carbohydrates. You're going to see that chemically speaking, they are simpler than their nucleotide or amino acid counterparts, where the nucleotides and amino acids have carbon, hydrogen, oxygen, nitrogen. And in the case of nucleic acids, you'll see phosphorus show up. Here, in the carbohydrate, you just have carbon, hydrogen, and oxygen. And they follow a very straightforward formula where the overall formula is represented here. You have a carbon and a water, and this repeats some number of times where N has to be equal to or greater than 3. Monosaccharides are also, they are aldehyde or ketone derivatives of these straight chain polyhydroxy alcohols that contain at least 3 carbon atoms. So you're going to see what this really looks like in a moment. Before I get to that point, though, I should emphasize that the most basic carbohydrate units, we are going to call those monosaccharides. There are various different types of monosaccharides, which are going to differ in the number of carbons and the arrangement of hydrogens and oxygens in terms of their attachment to the carbons. So they show up in different flavors, but generally speaking, they are still quite simple from a chemical perspective. The monosaccharides, just like any other biological molecule, can be combined to form different types of polysaccharides. So, for example, if you take 2 monosaccharides, put them together, you have a disaccharide. Take 3 monosaccharides and put them together, you have a trisaccharide. This can build into the oligosaccharide or polysaccharide language, just like when we talked about proteins. When we talked about proteins and peptides, you had oligopeptides, polypeptides, and so that language shows up here as well. Now if we look at two representative monosaccharides, you can get a sense for the structure here. So on the left-hand side, we have an example of an aldotriose. Okay, so what do I mean by that? Well, first, let's look at the number of carbons. 1, 2, and 3. Okay, so there's your tri. Now, any sugar, any monosaccharide that we're going to talk about is going to end with this ose language. So the triose tells me that I have a 3-carbon sugar. Now, if I look at the top here, the way that it is arranged at it's the top in this particular arrangement, I have an aldehyde functional group. Okay, so the aldehyde functional group is going to tell me that I have an aldotriose. So if the carbonyl group is at the end of the carbon chain, as it is in this example here, you have an aldehyde group at the end of your carbon chain. The monosaccharide is going to be classified as an aldose. But to be more specific, because we have 3 carbons, this is actually an aldotriose. For those that are curious, this is actually glyceraldehyde. You have seen glyceraldehyde before when we talked about the use of the DL language for amino acids. Glyceraldehyde is our reference molecule there. So here, now you get to see that glyceraldehyde is a relatively straightforward, a small representative monosaccharide. Now, remember, its classification as an aldotriose, or more broadly, just an aldose, is dependent upon the presence of an aldehyde functional group at the end of the carbon chain. So if I put this at the bottom, because of symmetry, it would effectively be the same thing, but at the end of the carbon chain, you have an aldehyde functional group, which classifies this as an aldose. It is also an aldotriose because of the 3 carbons, but I should really emphasize that not all aldotrioses are glyceraldehyde. You'll see in a moment that this comes back to the configuration of your carbons throughout. So I'll talk more about that in a moment. In contrast to the aldoses, if you have a ketone group, you have then a ketose. So on the right-hand side, you'll see a representative 3-carbon ketose. So 1, 2, 3 carbons. Now, instead of an aldehyde functional group at the end of the chain, you have an internal ketone functional group right here. And because this is a 3-carbon molecule, this is a ketose. So this is dihydroxyacetone. And so you'll see that glyceraldehyde versus dihydroxyacetone, these are both representative 3-carbon monosaccharides, but the real difference is where that carbonyl group is. Is it an aldehyde or is it a ketone? That would define one as an aldose versus a ketose. Now, while we are here, take note of all of the hydroxyl groups. So hydroxyl, hydroxyl, over here, hydroxyl, hydroxyl. So this is important because monosaccharides are freely soluble in water, but insoluble in nonpolar solvents. So these are very polar molecules. They can hydrogen bond with water very effectively. So very soluble in water. Most of them have a sweet taste. You probably have consumed monosaccharide, I don't know, sugar, table sugar, glucose. That's an example of a monosaccharide. So most of them do have a sweet taste. The backbones of your common monosaccharides are all going to be unbranched carbon chains in which all of the carbon atoms are linked by single bonds. So if you go through, you'll see you've got a backbone carbon chain, and all of your connectivity is through single bonds. Another theme of monosaccharides is that all monosaccharides, with the exception of dihydroxyacetone, contain one or more asymmetric chiral carbon atoms. If we look at glyceraldehyde, so you'll see a central carbon here. That carbon is attached to 1, 2, 3, 4 different things. So we have a chiral center right here. So what that means then is that glyceraldehyde, because it has one chiral center, it therefore has two different enantiomers, or two different optical isomers. There is a D form and an L form. So they are mirror images of one another that differ in configuration with respect to really the positioning, the absolute positioning of the hydroxyl and protons about this central tetrahedral carbon. This should really be no surprise to you. We talked about this when we were labeling D versus L for the amino acids. So here, because you have one chiral center, you have two different potential enantiomers. In general, a molecule with N chiral centers, N as in nugget, N chiral centers can have 2 to the nth stereoisomers. So if I have one chiral center, what that then means is that I have two different possible enantiomeric forms. Aldohexoses with 4 chiral centers, that's 2 to the 4th, have 16 different possible enantiomers, and you'll see monosaccharides with longer chain lengths, the configuration of your overall molecule is going to be defined based on the chiral center that is most distant from your carbonyl group. So later on, when we talk about the hexoses and the longer monosaccharides, the overall language of D versus L is defined based on the configuration of the chiral center that is going to be furthest away from your carbonyl group, whether it be the aldehyde or the ketone carbonyl functional group. Let's take a look at glucose as an example. So here I have the structure of glucose. You're going to see that there are 6 carbons. So what that means is that glucose first is a hexose. If we look at the presence of the carbonyl group, where's the carbonyl? Where's the carbonyl? Oh, it's right here at the end. You have an aldehyde group. So glucose is an aldohexose. Now if we look further, you're going to see 1 chiral center, 2 chiral center, 3 and 4 chiral center with a primary alcohol on the end here. So we have 4 chiral centers. So what that means is that we have 2 to the 4th, or 16 possible different stereoisomers or enantiomers. Now, when we look at glucose, you'll see here that the labeling is referring to D-glucose. So D-glucose here, the label says D-glucose down here. Okay, so I told you on the last slide that the overall configuration of the molecule is going to be defined based on the chiral center that is furthest from the carbonyl group. So here we have the carbonyl group at position 1. As we walk on down, you'll see, oh, at position 5, this is the chiral center that is furthest away from my carbonyl group. So as I look at this, in Fisher projection form, which is what is shown to you, I can basically take the configuration of this carbon and use it to label the overall molecule as being D. Now, again, much like we did for the amino acids, we're going to base this on glyceraldehyde. So if we look at the structure of glyceraldehyde over here, you'll see that in Fischer projection form, D-glyceraldehyde has the hydroxyl group at its lone chiral center pointed to the right. So we're not going to get into the, spatial positioning of the different functional groups that define the different stereoisomers. I just want you to be able to look at the Fischer projection and say, oh, okay, the hydroxyl group is pointing to the right at that chiral center, which defines this as being of the D configuration. If this hydroxyl group is pointed to the left, then you have an L configuration. So if we take that knowledge and translate it over to D-glucose, you should see at the fourth chiral center, or the chiral center that is furthest away from the carbonyl group, you have a hydroxyl group that is pointed to the right, which defines the overall molecules being of the D configuration. Now, I want to highlight here that if we walk through the different chiral centers, you'll see, oh, here, I have a hydroxyl group pointed to the left. So this individual carbon here, this chiral carbon, has a configuration that is defined as L. The first one, so carbon 2, would be defined as a D configuration, carbon 3, L, carbon 4, D, carbon 5, D. But we define the overall configuration based on this chiral center furthest away from that carbonyl group. So you've heard me say this a number of different times now. I'm trying to hammer this idea in that the overall configuration is defined based on the chirality, or the configuration at the chiral center furthest away from the carbonyl group. And this is true for all D sugars. All D sugars have the same absolute configuration at the asymmetric center furthest away from the carbonyl group relative to D-glyceraldehyde. So if ever in doubt, pull up the structure of L versus D-glyceraldehyde and make that comparison, and you will see that that is going to define the D sugar versus the L sugar. Now this is important. The stereoisomerism, the enantiomeric states of your sugar, is important because it is of great biological significance due to the fact that enzymes typically act on sugars in a very stereospecific way, where there is a preference for one stereoisomer relative to others. And there are cases where you'll see Km values, if you think about enzymes acting on a sugar as a substrate, we can measure how that occurs via Michaelis-Menten parameters. And if you look at Km values, you'll see that Km values will span more than 3 orders of magnitude, where the so-called incorrect sugar would bind much more weakly than the so-called correct configuration. You can think about this much like you would putting think back to the geometric complementarity that we talked about for enzymes. It's very much like putting a glove on your hand. So if you try to put a left glove on your right hand, it's not going to fit very well. So here, the stereochemistry, the stereo specific nature of how an enzyme binds to a substrate is going to be reflected here. So the overall configurations of your monosaccharide have very significant biological implications. If we look at the aldoses more broadly, you're going to start to see trends with regards to how we classify them. Let's start with the simplest case. So if we look at D-glyceraldehyde up here, first, it has 3 carbons, which defines it as a triose. There's also an aldehyde functional group, which defines it as an aldose. So here we have an aldotriose. Now, if you look at this molecule, you'll see there's a single chiral center. So here you can either have D-glyceraldehyde or L-glyceraldehyde. That's it. That's really the simplest way, the simplest grouping that we have. Now, let's imagine a situation where we start growing the monosaccharide. We start inserting additional chiral centers. So let's take glyceraldehyde and let's throw in a new chiral center between the aldehyde group and the original chiral center right here. Okay, so that gives us 2 new monosaccharides. Remember here, I want to highlight that we inserted a new chiral center between the aldehyde group and the original chiral center. So that's what gave us these 2 new monosaccharides. Now, the insertion of a new chiral center, this could occur with 2 different configurations. It could be L, could be D. So that configuration is what defines the identity of these 2 new monosaccharides. Now, by adding in a new carbon, we have expanded. We are no longer looking at a triose. We are looking at tetroses. So now we're looking at the aldotetroses, in fact. We still have that aldehyde group. So we have an aldose. It has 4 carbons, 1, 2, 3, and 4. So this makes for an aldotetrose. But the configuration of the newly inserted chiral center defines D-erythrose versus D-threose. If you look at this, you should see here that in the case of D-erythrose, your hydroxyl group is pointing to the right, so this carbon has the D configuration, whereas in D-threose, the hydroxyl group is pointing to the left. And so this carbon would be defined as having an L configuration. So at carbon 2, so carbon 1, carbon 2, 3, and 4 at carbon 2, that configuration distinguishes the members of each grouping of monosaccharides. Now, these are both still D sugars. If you look at the chiral center, so that would be this chiral center or this chiral center, they retain the original configuration of D-glyceraldehyde. And remember, the configuration of that chiral center that is furthest away from the carbonyl group defines the overall configuration of the molecules. So here, by having a D configuration, also a D configuration over here, my sugars, erythrose versus threose, here are defined as being of the D configuration. Okay, so for glyceraldehyde, we had 1 chiral center. So 2 to the 1th meant that I had 2 different possible enantiomers. Now we have 1 and 2. Okay, 2 chiral centers, so 2 raised to the 2nd power. That is 4. So for the aldotetroses, as shown, we have 4 different possible enantiomers. We would have D-erythrose, L-erythrose, D-threose, L-threose. These are options. But now, let's keep on going. Let's insert another chiral center, another new chiral center between the aldehyde and that carbon 2 position for the tetroses. So if we move on down, we do this, we've inserted a new chiral center, and now you'll see that, again, we have an expanded possible arrangement of configurations, a number of expanded possibilities for enantiomeric combinations. So if we took erythrose and did this, that newly inserted carbon could be inserted with the D configuration or the L configuration, and the other groupings are retained. So here you'll see these are both D configurations at 1, 2, carbons 3 and 4. These are retained from D-erythrose, whereas over here for threose you had an L and a D, so L, D. So you'll see how threose or erythrose, how this sort of propagates downward, but the newly incorporated chiral center can occur with only 2 different configurations, which defines ribose versus arabinose and xylose versus lixose. But yet again, the configuration around C2 distinguishes the members of each pairing. So you can distinguish xylose versus lixose based on the C2 configuration, likewise ribose versus arabinose. Okay, but yet again, you'll see that the chiral center furthest away from my carbonyl group is always D in this grouping. So D-ribose, D-arabinose, D-xylose, and so on. You can sort of see how this goes. If we do this yet again, we're going to go from the aldopentoses to the aldohexoses, and you can see that the complexity just grows. And remember, if we look at the number of chiral centers, so 2 to the nth, that is going to tell us the number of possible enantiomers. So 1, 2, 3, 4. 4 chiral centers for a typical aldohexose gives me 16 different possible enantiomers. Now, before I end here, one thing I want to go ahead and point out is, it is common for students at this point to look at this table and say, oh no, I have to memorize this. No, you don't. What I want you to be able to do is look at monosaccharides and tell me, let's say I give you a monosaccharide pairing. Let's say I throw up ribose and arabinose. What I want you to do is look at those molecules and tell me, first, how would they be related? And you should see that ribose and arabinose are related through that C2 configuration. So you should be able to tell me that. And tell me the overall sugar configuration, not necessarily what it is. So I don't necessarily need for you to be able to tell me, oh, that's galactose versus glucose. I'll give you that information. But you should be able to look at the configurations, look at the functional groups, and sort of tell me something about the molecule. Everything that I just told you for the aldoses can be readily translated over into the ketoses. The difference is, instead of starting with glyceraldehyde, we're going to start with dihydroxyacetone. Now, same sort of story. Glyceraldehyde is a 3-carbon aldose, so an aldotriose. Dihydroxyacetone is a 3-carbon ketose, or a ketotriose. Now, unlike the aldoses, the ketoses are defined based on the C3 configuration. So remember, when we started trying to differentiate the different pairings, I told you that for the aldoses, it was the C2 chiral center that differentiated, but here it's C3. Now, remember, dihydroxyacetone does not have a chiral center. So how does this start showing up? How do we differentiate the enantiomers? Okay, so there's only one kind of dihydroxyacetone. There's no stereochemistry that you have to think about. But if you were to take a chiral center and insert between this carbonyl group here and this primary alcohol down here, you would get erythrulose. Now, you'll already notice when we talked about the aldoses, there was an erythrose. Well, here, for the 4 and 5 carbon ketoses, we differentiate the aldose from the ketose by inserting UL into the name. So erythrose becomes erythulose, which is what is shown here. Ribose becomes ribulose. Xylose becomes xylulose. And so you can sort of see easily for the 4 and 5 carbon ketoses how you would differentiate the aldose form from the ketose form. Okay, so as we go, you'll see the same sort of logic applies to the ketoses as we saw for the aldoses. You have 2 to the nth number of different possible enantiomeric forms that are based on the number of chiral centers. So for erythulose, you have 1 chiral center right here. It is of the D configuration, which defines the overall molecule as being of the D configuration. So that's why this is D-erythrulose. And so we have 1 chiral center, 2 to the 1th, 2 different possible enantiomers. So we have possible D and L forms for this molecule. Now, again, if we insert another chiral center between the carbonyl group and this chiral center, that then gives you additional enantiomeric possibilities. So for ribulose, for example, which is a 1, 2, 3, 4, 5 carbon molecule, you have 2 different chiral centers. So 2 to the nth, 2 to the 2nd, that gives you 4 different possible enantiomeric forms here. But yet again, just like the aldoses, the overall molecule's stereochemistry is defined by the chiral center that is furthest away from the carbonyl group. It just so happens that in this case, the carbonyl group is a ketone, not an aldehyde group as you saw for the aldoses. Now, once you get beyond the 4 and 5 carbon molecules to the ketohexoses, you'll see the names diverge. We don't have the same sort of naming system, which really allows for direct comparison between aldose and ketos. Here you have more case-specific names. You probably all heard of fructose at some point, high fructose corn syrup being the great example of that. So fructose is a 6-carbon ketohexose. So yeah, going forward, you'll see examples where we will talk about the ketoses versus the aldoses, but now you should have an appreciation for what differentiates one from the other. Earlier when I showed you the aldoses, you might have noticed that you had sugars that were very closely related to one another, but really differed only in configuration and at a single carbon group. So these are called epimers. Sugars that differ only by configuration around one carbon atom are called epimers. So as an example, let's look at glucose. So glucose is an aldohexose. You have 6 carbons and an aldehyde group here at the end, which defines it as an aldohexose. Overall, as shown, this is D-glucose, which is based on the configuration at this carbon 5, which is the chiral center furthest away from your carbonyl group. Now, let's just say we flip the configuration at carbon 2. We take this carbon, which currently in glucose has the D configuration, and we convert it over to the L configuration. Okay, at that point, we no longer have glucose, we actually have mannose. So you should see here that D-mannose and D-glucose are related only by, or rather they differ only by a swap of configuration at the C2 chiral center. So mannose, D-mannose, and D-glucose are epimers. They are in fact referred to as C2 epimers. Okay, so while I'm at this mannose-glucose comparison, let's say we compared L-mannose with D-glucose. These would not be epimers because then you would have, so in order for D-mannose to be converted to L-mannose, you would have to convert the configuration at carbon 5 while retaining everything else. So what that would then mean is if you compared it to D-glucose, you would differ in configuration at carbon 2 as well as carbon 5, which would not satisfy the definition of an epimer. Sugars that differ only in configuration around one carbon atom are epimers. So D-mannose and D-glucose are C2 epimers, but L-mannose and D-glucose would not be epimers. So just take note of that. Similarly, D-glucose is a C4 epimer of D-galactose. So if you look at all of your carbon atoms, so your C2 group is D, okay, in galactose it's also D, C3, L, same thing in galactose, but at carbon 4, you have an inversion of configuration. In glucose, this is a D chiral center. In galactose, this is L. And likewise, at carbon 5, they are both of the D configuration, which defines the configuration of the overall molecule. So D-glucose and D-galactose are related to one another in the configuration at C4. They differ in configuration at C4, which makes them C4 epimers. Yet again, D-glucose and L-galactose, on the other hand, would not be epimers because they would differ in configuration at 2 chiral centers. So remember, the epimer has an epimeric pair can only differ in configuration around one chiral atom. Up to this point, we have only looked at the sugars, the monosaccharides, in their linear open chain form. However, you will notice that you have functional groups in those straight chain forms that are reactive with one another. Principally, if you look at the reactions of alcohols with carbonyl groups, you're going to see that is going to drive very particular behaviors in monosaccharides. So let's just take a look at that here. So alcohols react with the carbonyl group of either aldehydes or ketones. So the alcohol is going to act as a good nucleophile and undergo nucleophilic attack on the electrophilic carbon of either the aldehyde or ketone functional groups. The product is going to be either the hemiacetal, which is a tetrahedral carbon with substituents as shown, or the hemiketal. Again, that product has a tetrahedral carbon with substituents as shown, where really the difference is one R group or two R groups. That is really the difference between hemiacetal and hemiketal. And you should see sort of where the substituent R groups come from. If you start with an aldehyde, you would have a hydrogen atom, and that would be the resulting, you'd still have that hydrogen atom over here. If you had a ketone as your starting reactant, you have an R1 and R2 substituent, which is still there in the hemiketal form. Okay, so as you'll see in a moment, this is going to drive certain structures for the monosaccharides. And let's just go ahead and hop into that. So let's hop into an example. In the upper panel, you're going to see the example of D-glucose. So D-glucose being an aldohexose. In the bottom panel, you're going to see the example for D-fructose, which is a ketohexose. Okay, so remember, we're looking really at the reaction between alcohol and carbonyl. In the case of D-glucose, what you can and do see is reaction of your C5 alcohol with your aldehyde. So there's enough flexibility here where this can sort of wrap around, and the C5 alcohol can undergo nucleophilic attack on that carbonyl, that aldehyde group at your carbon 1, your carbonyl carbon. This is going to then drive closure, ring closure, to give you a closed ring form of glucose. And as shown, this would be what we would refer to as a pyranose ring. So as shown, this is beta D glucopyranose. And we'll talk about where the beta language comes from in a bit. But for the moment, just know that this is D glucopyranose, where it is a glucose based on the configuration of the different chiral centers. It is defined as being glucose, and it is a pyranose ring, which is overall going to be a 1, 2, 3, 4, 5, and 6-membered ring. You might ask, why can carbon 4, that hydroxyl group, not do the same thing? Well, it can, but for glucose, it doesn't do that quite so readily. For carbons C3 and C2, sterics really prevent that from happening. But if you look at the resulting glucopyranose ring down here and place emphasis on carbon 1, what you should note is that this is a hemiacetal functional group. You have, if you draw the comparison here, here's one oxygen, here's another, and then here's your proton, and your R group is carbon 2. So you have a hemiacetal group right here at carbon 1. Now, if we do the same thing for D-fructose, your chemistry is not occurring, your carbonyl group is not at carbon 1, it's actually at carbon 2. So that's shown in blue down here. Again, your C5 alcohol is going to undergo nucleophilic attack on that carbonyl group, that carbonyl carbon, to give a closed ring form yet again. Except now you have a 5-membered ring, not a 6-membered ring. So the 5-membered ring is defined as a furanose ring. Remember, the 6-membered ring is the pyranose form. The 5-membered ring is the furanose form. And in this case, the ring closure generates a hemiketal. And this is all based on the fact that you started with a carbonyl group that was in the ketone form, not the aldehyde form. So you're going to see momentarily that the sugars can exist in both the linear open chain form as well as the closed ring form, and that the conversion between the two is readily reversible. So you have the double arrow structure shown here, ring closure, ring opening. This occurs in a constant back and forth. So you're going to see that this readily occurs in solution, and that usually you're actually going to have a mixture that establishes an equilibrium between the two states. I've just introduced this language to you, but to really hammer the idea in, a sugar with a 6-membered ring is known as a pyranose, and a sugar with a 5-membered ring is designated as a furanose. So why are we focusing on these? Well, in principle, hexoses and larger sugars can form rings that include 7 or more atoms, but these rings are rarely observed because the 5 and 6-membered rings are just simply more stable. So the pyranose and the furanose forms happen to be the more stable ring forms. In contrast, 3 and 4-membered rings experience great internal strain, and that also makes them less stable than the linear form. So when you see the closed ring forms for monosaccharides, you are almost always going to see either pyranoses or furanoses. Towards that point, here, if we look at D-glucose versus D-ribose, you're going to see that both monosaccharides can form pyranose or furanose forms. So for D-glucose, if we had an attack from 1, 2, 3, 4, the C4 alcohol on the carbonyl group, you would then get the furanose form, which would be your 5-membered ring, or if you had the attack, the nucleophilic attack from the C5 alcohol on your aldehyde, you get the pyranose form. You'll see a little bit later that for D-glucose, the pyranose form is actually more stable than the furanose form. So just because you can doesn't mean that you always do. The same story is true for D-ribose as well. So you can form the pyranose or furanose forms, and this is all dependent upon whether or not you have a nucleophilic attack from your C4 or C5 alcohol. The nucleophilic attack to generate the hemiketal or hemiacetal functional group, as we just talked about, that basically that ring closure event. This process generates in the hemiketal or hemiacetal a new chiral center that itself has 2 possible configurations. So if you look at the functional groups that are undergoing a reaction, in this case you're looking at D-glucose, so here you have your C5 alcohol group undergoing nucleophilic attack on that C1 carbonyl carbon. Okay, so this can occur in 2 ways. This could be a front side or back side attack. So the hydroxyl group could approach from the front or from the other side, from the back side. This is going to dictate 2 possible resulting configurations for that aldehyde carbon, or what was the original aldehyde carbon. Now, for this reason, we refer to this carbon as the anomeric carbon. So in the closed ring sugar, the anomeric carbon is always designated as C1. And so here you'll see that you have 2 different forms. You have the alpha form and the beta form. Really the difference here, if you want to think about this just in qualitative language, the alpha form always has this substituent hydroxyl group pointed down, and you can think about this as being down below the plane of the sugar, whereas the beta form points up. So you'll see this play out more when we talk about nucleotides and other things later, but the front side versus back side attack gives you 2 different possible configurations for that anomeric carbon, one of which positions the hydroxyl group, or rather the substituent, down to give you the alpha form, and the other points that substituent up to give you the beta form. Now, we have a 6-membered ring that is generated from glucose. So this is glucopyranose, not only glucopyranose, but we started with D-glucose, so D-glucopyranose, and now we have an alpha D-glucopyranose possibility versus a beta D-glucopyranose possibility. So the alpha versus beta really tells you whether or not you had a front side or back side attack, and the rest of the language tells you something about the sugar itself. Let's look at glucose again. So D-glucose, it can exist in alpha or beta or linear forms. So if we look at the alpha form, just as an example, We just talked about this, but I want to expand on it a little bit. So I told you that the alpha versus beta is designated on the basis of the position of that C1 alcohol substituent. If it's pointed up, it's beta. If it's pointed down, it's alpha. But how do you know which way is up or down? What if we have mistakenly oriented the molecule in a weird way? Well, your benchmark is the positioning of the C6 alcohol. So if you look at the C6 alcohol over here, as shown, it is pointed up above the plane of the sugar. For the alpha form, that C1 alcohol substituent is pointed down below the plane, and you know that it is below the plane because the C6 alcohol position designates the above plane, where that is. So the C1 alcohol is pointed in the opposite direction as the C6 alcohol in the alpha form, but in the beta form, they're pointed up in the same direction. Okay, so D-glucose. I'm showing you the alpha form, I'm showing you the linear form and the beta form, but what is real here? What actually exists? Why should you care about them all if they don't all exist? Well, the alpha and beta anomers, as we will call them, of D-glucose, they interconvert in aqueous solution by a process called mutarotation. Mutarotation is a process wherein one ring form opens briefly into the linear form and then closes again to form the other form. So this is readily reversible, going from alpha form to linear to beta and then back. This is pretty reversible. So a solution, let's say I started with a solution of pure beta D-glucose, and I also had a solution of pure alpha D-glucose. Let's just say I had them separately. Eventually, what you're going to see is that you're going to form for these very different starting points. Remember, in one case we had pure alpha D-glucose and the other pure beta D-glucose. Because this is a reversible equilibrium, they're going to, if allowed to incubate long enough, they will reach the same point. They will form an identical equilibrium. And what that equilibrium looks like is this. The mixture is going to consist of about 1/3 alpha D-glucose, 2/3 beta D-glucose, and very small amounts of the linear and 5-membered ring forms. So the dominant species here is going to be the beta D-glucose. And remember, I just gave you the example of starting with pure beta D-glucose versus pure alpha D-glucose, and you reach the same equilibrium mixture. So the same mixture is gotten regardless of starting point, because this is an equilibrium process. So once you reach equilibrium, you have a mixture of these species where the linear form actually populates very little. It's mostly alpha, so 1/3 alpha and 2/3 beta D-glucose. Though I have been using the language in previous slides describing the sugar as forming a planar structure, that's not technically correct. The 6-member Pyrenees ring is not planar. So you should remember some of this from organic chemistry. Instead of being planar, the Pyrenees ring actually adopts possible chair and boat conformations. Now remember, the conformation of a molecule, if you have multiple conformations, you can interconvert without breakage of covalent bonds. This is basically a rotational relationship. Configurations, on the other hand, must involve bond breaking. So configurations require you to break bonds. So going from, say, for example, the beta to the alpha form requires a bond to be broken. But to go between conformations requires rotational alterations, which do not require bond breakage. Now, when we look at the beta D-glucose, or even the alpha D-glucose shown here, but let's focus on the beta D-glucose, you have two different possible chair conformations. These are not readily interconvertible. You're going to see that there's about a 46 kilojoule energy barrier that separates them. So you have to put in about 46 kilojoules of energy to convert one to the other. So this interconversion does not readily occur. The boat conformation is really only seen in derivatives with bulky substituents. So when we look at this, the common chair forms for beta D purinose, you're going to see that of the two chair conformations, the one that predominates is the one in which the bulkiest substituents occupy equatorial positions rather than the more crowded axial positions. So there is, even though you have two different conformational possibilities, there is a preferred conformation that really just positions substituents to minimize steric clash. A monosaccharide, it has alcohols, it has aldehydes, ketones potentially. So what that means is that these functional groups, this is not a special situation where they stop doing the normal chemistry that they would be doing, in other molecules. For that reason, the cyclic and linear forms of both aldoses and ketoses can interconvert, and this can involve reactions that are more typical of aldehydes and ketones, which ultimately leads to modifications to your monosaccharide structure. So we're going to go through some example modifications here very briefly. The first of which is just simple oxidation of your aldehyde group to a carboxylic acid. So here, this C1 group would normally be an aldehyde. If you oxidize that to a carboxylic acid, you now have something called an aldonic acid. So this is not an aldose anymore. It is an aldonic acid. Aldonic acids are named by appending the suffix onic acid to the root of the parent aldose. So if you started with D-glucose, now you have D-gluconic acid. In contrast, let's say we oxidize the C6 alcohol on the other end. So in the first example, we oxidized that aldehyde group, but now we are looking at oxidation of the C6 primary alcohol that was there. So in this case, we took the alcohol and oxidized it to the carboxylic acid, much like we did in the previous example. So now we are going to just name this to reflect that. So instead of this being glucose, now it is D-glucuronic acid. So here, when you do this, when you oxidize that C6 alcohol for any of your monosaccharides, you get what is called a uronic acid. And so we name accordingly by appending the uronic acid to the root name of the parent aldose. So D-glucose is D-glucuronic acid. Reduction is also a possibility. So you started with, in most cases, an aldehyde containing polyalcohol. So you have starting, let's say you started with ribose or xylose or glyceraldehyde. All of those had an aldehyde functional group at the C1 position. But let's say we reduce that aldehyde into an alcohol, and now we just have a polyhydroxy alcohol that lacks that carbonyl group. So in this case, we are going to change the parent name, the root name, to have the etol ending. So ribose becomes ribitol, xylose becomes xylitol, glyceraldehyde became glycerol. You're going to see examples of this if you ever go out and look for artificial sweeteners. You'll find that these polyalcohols are very common artificial sweeteners, where ribitol and xylitol are common substitutes for, say glucose or, other more traditional sugars. Other modifications include substitution of OH groups. So a very common example that you will see in the next chapter is substitution of an OH group with a hydrogen. So generally, these are referred to as deoxy sugars. So here you are looking at the example of beta D 2-deoxyribose, where normally you would have an OH group right here, but it has been substituted with a hydrogen. So here, these deoxy sugars represent the basis for many nucleotide structures that we'll talk about in the next chapter. We also have a grouping referred to as amino sugars, where one or more OH groups are replaced by an amino group. So here, D-glucose, actually alpha D-glucose, has been subjected to the same sort of modification to give alpha D-glucosamine. Here is alpha D-galactosamine. So here you had an OH group that was replaced with an amino group to give the amino sugar. And often, actually, you've already seen an example of this, but often you're going to see that these amino groups are acylated. We saw an example of this when we looked at peptidoglycan. We had an acylated amino group there that represented a potential point of conjugation to other molecules. So amino sugars are very common as well. In this final slide, we have a general representation of common modifications that are made to sugars, where, let's say we start with glucose, you can see the generation of amino sugars or deoxy sugars, acidic sugars. You can see phosphorylated sugars too. So there are a number of different modifications that are possible and usually are made as the need arises. One major example of this is the phosphorylated sugar. Most cells do not have plasma membranes that allow for the transport of phosphorylated sugars. So once you phosphorylate a sugar, the effect is basically the sugar becomes trapped inside of the cell. This is important because the example shown here, beta-D-glucose 6-phosphate, this is the first molecule that you see show up in glycolysis. And I'm giving you this as an example because what I want you to understand is that once we start doing metabolism, we are investing energy into this molecule to ultimately downstream make ATP. We wouldn't want to invest energy into preparing this molecule towards that goal if this molecule were just going to simply diffuse out of the cell. So by phosphorylating it, we basically trap it in the cell, and we can then know that our investment is going to stick around. So you'll see other examples of things like this, but the modifications are really generally going to center on the need. Why, what physiologic need is there, and then what modification can facilitate us reaching that need. Okay, so this concludes our discussion of monosaccharides. Next time, we're going to discuss polysaccharides, and you will see that we will start looking at unique combinations of the building block monosaccharides that have been modified in different ways, and ultimately this is going to give a broad range of different properties that are going to be associated with your different monosaccharides that once built into the polysaccharide.